Hello, my name is Calli Nguyen. I am currently a rising senior and undergraduate researcher at Louisiana State University in Baton Rouge. And this summer, I've had the esteemed pleasure of working in Dr. Bolin Liu's lab at the Louisiana Cancer Research Center, with Dr. Hui Liu being my mentor. Today, I'll be talking to you about my study on the identification of biomarkers and effective treatments against Herceptin-resistant breast cancer. Herceptin resistance does remain a challenge in the successful treatment of HER2-positive breast cancer, with breast cancer being the leading cause of cancer-related deaths in American women, with overexpression being observed in about 20 to 25% of all breast cancers. There is a lack of efficient biomarkers for Herceptin response indication, with Herceptin resistance being one of the many clinical challenges. Novel therapy overcoming Herceptin resistance can be promising for the reduction or elimination of mortality related to Herceptin-resistant breast cancer. And the main aim of our study was to identify biomarkers and develop possible epigenetic approaches with therapeutic potential. In order to do so, Herceptin-resistant cultures of HR20 and POL2 were utilized, and these are two well-known HER2-positive breast cancer cell lines that are sensitive to Herceptin. Multiple analyses via Western blot and cell proliferation MTS assay were performed to detect specific proteins and determine cell sensitivity to Herceptin respectively. Regarding the MTS assay, the cells were seeded on 96 well plates with 4,000 cells per well with varying concentrations of Herceptin, panabino, and a combination of the two drugs. On day 2, these cells were incubated and the absorbance was reported. And regarding Western blotting, the REPA buffer and a cocktail of halt protease and phosphatase inhibitor were used in conjunction with the appropriate measurements from the BCA protein assay kit to extract the total protein from the cells. And then gels were run and a transfer process was performed to obtain the membranes, which were then blocked with 5% non-fat milk in a tris buffered saline with incubation with a primary and a secondary antibody. Enhanced chemiluminescence was then performed to detect the signal. And herein we disclose the effects of a potent anti-proliferative pathway for overcoming Herceptin resistance. Panabino is an inhibitor for histone deacetylase or HDAC. A combination drug treatment was shown to enhance the overall induction of apoptosis, which can be shown in figure 1. We see that a single treatment of either drug can show promise, but that there are more additive effects in cell growth inhibition with a combination of the two drugs. Similarly to the Western blot comparisons, figure 2 shows that my data using HR20, whereas figure 3 showing my brilliant mentors data with POL2, we see that the protein expression and how these cleaved lines represent the apoptotic effects that the treatments had. We see how a combination of the two drug treatments show additive effects in cell growth inhibition as well. In figure 2, panabino enhances Herceptin, which induces apoptosis, and likewise in figure 3, with panabino enhancing lepidinib. And from these results, a possible target to study and experiment more into was HDAC. And as previously stated, panabino is an HDAC inhibitor, and this is due to its ability to increase PBB3CB, which then increases FOXO3A by increasing its expression, promoting expression of miRNA, and then finally inhibiting IGF-2 via a decrease in expression. And in other words, PBB3CB, FOXO3A, and the IGF-2 signaling contributed to a positive response of Herceptin-resistant treatments. In conclusion, while this study has promise for possible treatments against Herceptin-resistant breast cancer, the precise mechanism does remain elusive. And we suggest that further clinical evaluation could be implemented. The study does provide new avenues to identify other potent biomarkers for predictive indication of overcoming Herceptin resistance. These results could indicate a promising method for the reduction or the elimination of mortality related to Herceptin-resistant breast cancer. Then lastly, I would love to take this time to acknowledge and personally thank my PI, Dr. Bolin Liu, and the Department of Interdisciplinary Oncology, Stanley S. Scott Cancer Center, the School of Medicine, the LSU Health Sciences Center in New Orleans, my intelligent mentor, Dr. Hui Liu, for her guidance, expertise, and leadership, the National Institutes of Health, National Cancer Institute, and the Cancer Research Education Program for funding, and finally, my audience for taking the time out of your busy schedules for listening to my research. And I'll now open the floor to any questions or comments you may have. Thank you so much.